we're going to take a look at some of the main points of respiration, cellular respiration specifically. This is going to be happening in the mitochondria. The matrix is the inner liquid, sort of cytoplasm-like substance. There is an outer and an inner mitochondrial membrane that is going to be important with something called chemiosmosis. The cristae are the finger-like projections that sort of stick out into the matrix. And inside that, we've also got 70S ribosomes, just because these guys came from prokaryotes a long time ago. So the equation for cellular respiration is C6H12O6, or glucose, plus approximately 6 oxygens. Through the process of cellular respiration, which is outlined up here, is going to become 6 molecules of carbon dioxide, 6 molecules of water, and a whole bunch of other small byproducts in your body that hopefully yield you a lot of ATP, because that's how you run. This is a summary page. We want to get 2 ATP out of glycolysis. We want to get 2 ATP from the Krebs cycle, and hopefully about 32 ATP from the oxidative phosphorylation near the electron transport chain, yielding you 36 ATP. So when we look at glycolysis, which is the first step that happens in the cytoplasm of the cell, you start off with a 6-carbon or hexose sugar. Now you've got to use some ATP here in order to change it into its next structure, which can be seen here. Phosphates are added from the ATP that we use. Then we split that molecule apart and we oxidize it, as you can see here. And what we want to get ultimately is this molecule NAD plus turned into NADH. That's going to come in handy a little bit later. Now, doing this process outlined right here is actually going to yield a few ATP for us. And what we have at the end are the pyruvate molecules you can see down here, 2 3-carbon molecules from 1 6-carbon molecule earlier. Using 2 ATP and producing 4 ATP yields us 2 ATP from glycolysis in the cytoplasm. From here, something called oxidative decarboxylation can happen. Our pyruvate is going to be split up by a little coenzyme called acetyl-CoA. Now again, this is going to do some more oxidation and provide us with NADH, which again will come in handy in the electron transport chain. After this, what we have are 2 of these carbon molecules attached to this acetyl-CoA molecule. And we also use 1 of those carbons to accept some oxygens in the form of CO2, which helps fill out the equation that we were looking at before. Now that we're in the Krebs cycle, it looks complicated, but just keep in mind what we have up here. What do we need to make? How many of them do we need to make? That's really the main point here. The diagram's just details in the middle. So, from 1 glucose, we are going to add a couple of carbons from acetyl-CoA onto a 4-carbon molecule that resulted from the Krebs cycle. That creates a 6-carbon molecule here. That 6-carbon molecule is going to be rearranged, and as that happens, we make an NADH. The next molecule that we can see is going to, again, 5 carbons, be rearranged and make another NADH. Both times we did that, we also made a CO2 molecule, again, helping us out with the equation before. What we then get is, from this 4-carbon molecule, a slight rearrangement of its structure yields us with a single ATP. We also get FAD turned into FADH2. And once again, we get NAD plus turned into NADH. Now, that was only for 1 of the 2 acetyl-CoAs that we're ultimately going to get out of this. But if you remember in this last process, there were 2 pyruvates, which means we get 2 acetyl-CoAs, which means we do this process 2 times around. If you do that and you count it up, we're ultimately going to get not 1, 2, 3, but 6 NADH. And we're not going to get just 1 FADH2, we'll get 2 of them. And we won't just get 1 ATP, we'll get 2 of them per glucose that we started out with. That was the trickiest part. From here, we need to move, that last part was in the mitochondria, in the matrix. Here, we're going to also use the inner mitochondrial membrane. NADH is going to be reduced. It's going to lose its hydrogen and some oxygen, or sorry, and some electrons. And that proton is going to move into the intermembrane space, the space between the inner and outer mitochondrial membrane. Same thing for FADH2. It is reduced, it loses its hydrogen proton, electrons are lost as well, and those go into the intermembrane space. This is creating a concentration gradient of hydrogen protons. Now at this point, this concentration gradient will flow through my personal favorite protein enzyme, ATP synthase, in something we call chemiosmosis. This flow of hydrogen protons through ATP synthase generates a huge yield of ATP, 32 to be exact. Now, some of these hydrogens and electrons need to be mopped up or used, and so we can actually get rid of the excess ones with a little enzyme here in the membrane that's going to take a couple of hydrogens and electrons and combine them with the oxygen you breathe to actually make a little bit of water in your body. So this summarizes where these processes happen. Glycolysis is in the cytoplasm. The Krebs cycle and the electron transport chain are inside of various parts of the mitochondria. In addition, if you don't have any oxygen available, you will start doing what is called anaerobic respiration. And that uses no oxygen, but it doesn't get you as much ATP. There is CO2 and ethanol produced if you are a yeast, or if you are animals, you might make lactic acid. And this whole thing will occur in the cytoplasm. Mitochondria are not used for it at all.